Hi, I'm Lauren Henke, and I'm here with my co host, Stephen Chun, back for day two of ASCO 2026. Today, we bring you a translational study looking at the gene profile of high risk prostate cancer over the span of decades. The lead of the study is Dr. Krishnan Patel, an associate professor of radiation oncology, recently recruited to the University of Texas MD Anderson Cancer Center.
Dr. Patel, welcome to the NRG Oncology Podcast, and congratulations on presenting these exciting findings at ASCO 2026. To start off, this is an analysis of RTOG trials that are in some cases over 30 years old when radiation and combined modality therapy were in their infancy. In fact, the most recent trial in this analysis is RTOG 0521, which was launched over 20 years ago looking at adding high-dose docetaxel for high-risk prostate cancer. Can you start off by explaining why these decades-old studies are still relevant to us in 2026 and why a heterogeneous set of trials made sense to conduct this analysis?
Thanks so much. It's a pleasure to be here. This is a great question. The study of prostate cancer, unlike other malignancies such as lung cancer or glioblastoma, is a disease which can require much longer follow-up to gather enough events to study reliably. As you mentioned, this study includes data from 4 trials, the most contemporary for which was RTOG 0521, a randomized trial of radiation with or without docetaxel. This study had a primary objective to demonstrate a survival benefit from intensification with docetaxel. This was a trial which accrued between 2005 and 2009. The initial report, with a median follow-up of about 6 years, was in 2019, and the long-term follow-up, with a median follow-up of about 10 years, was published in 2023. At this time, only about 1/3 of patients had died, and only about 40% of these deaths were due to prostate cancer, which was really the signal that investigators were hoping to measure. So, to answer your first question, as you can see, given the long natural history of prostate cancer, it naturally may take decades from study initiation to allow us to measure hard endpoints. Pertaining to the second part of your question, the heterogeneity of patients profiled in this study is an important contextual feature, so thank you for allowing me to expand on it. From my perspective, heterogeneity can both be a blessing and a curse. You could, for instance, imagine a study which enrolled a very narrow group of patients, all of the same age, T, N, and M stage, and grade of localized prostate cancer. Such a study would result in very precise conclusions about the subgroup of patients under study, but would have relatively poor generalizability. On the other hand, you could design a study with very broad inclusion criteria. Take as an extreme example, a study which enrolled any patient with any stage or histology of any cancer. As will be obvious to any clinical oncologist, such a study would obviously be too broad to capture any meaningful conclusions to apply clinically. So, it is obvious that there's a sweet spot somewhere between the two extremes outlined in these examples, but it's often unclear exactly where this sweet spot lies. In respect to other prior studies on the topic which have combined both post-surgical and post-radiation patients to evaluate long-term outcomes, some may consider the trial relatively low on this axis of heterogeneity. We do attempt to address this in 2 ways. One, by evaluating only the subgroup of NCC and high or very high-risk patients from these trials, in which it would make most sense to consider abiraterone intensification. And 2, by utilizing adjusted models to adjust for differences in patient characteristics and randomized treatment between the trials.
Transcriptomics can be quite opaque, especially to people who don't do basic science research. What are transcriptomics, and how does the 22 gene decipher test work for prostate cancer? Am I correct in my understanding that the decipher is similar in principle to the Oncotype DX test that we use often for breast cancer?
I agree. With the dizzying array of advanced biomarkers, it's sometimes hard for us as clinicians to keep up with the details of these frontier tests. In summary, transcriptomics is the characterization of the transcriptome, or messenger RNA, which is transcribed from a tumor cell's DNA. This sort of assay gets information that is one step closer to the eventual effector proteins than an analysis of the DNA itself. And so, it is felt to represent information closer to the actual phenotype of behavior of the tumor cell. It also gives us some picture of the levels of gene expression. So, with that 20,000-foot overview, Decipher does use a similar principle to the Oncotype DX test for breast cancer. The commonality is that they both profile mRNA, although with slightly different technologies. Oncotype DX uses qRT-PCR to perform a very targeted look at 21 genes. In contrast, Decipher uses a microarray, which evaluates the levels of mRNA from many thousands of genes. A model is then used to specifically combine 22 genes out of the thousands assayed to produce a single score.
With this all out of the way, What were the aims of this study, and how was Decipher used to achieve these goals?
The objective of our study was relatively simple, and one born from a clinical scenario which seems to come up more and more nowadays. We wanted to understand if and how we could combine the standard NCCN risk stratification model with the 22-gene genomic classifier score to determine a better way of assessing patients for abiraterone candidacy than mere clinical judgment alone. We had three main objectives here. First, to evaluate if the genomic classifier score could improve prognosis over standard clinical variables alone, for example, PSA, Gleason score, stage, etc. And we found that it could. This helped to justify combining the two into a single model. Second, we then sought to understand if discordance between clinical risk stratification and biomarker risk stratification often occurred. This was important because if, for instance, we had found that 99% of the time the NCCN risk group and the Decipher risk group both agreed, there would be little added clinical utility to combining the two. We found that discordance happened approximately 25% of the time, which we felt was clinically meaningful and reflective of modern practice. Lastly, we wanted to generate a simple parsimonious system that clinicians could use in their clinics to help select patients for abiraterone intensification, and that was the main innovation in our study.
So, what were the major paradigm-changing findings that you got to present today at ASCO? And what is the potential impact on patients out there who might be facing a high-risk prostate cancer diagnosis?
The clinically relevant finding was the development of this simple algorithm to help physicians decide on candidacy for abiraterone intensification when combining NCCN risk stratification and genomic risk stratification. Although a figure summarizes the system much more clearly, I will attempt a narrative summary for our podcast audience. To use this system, you calculate a total score for each patient, which is the sum of two scores. The first score is based on the NCCN risk stratum, and the second is based on the genomic classifier risk stratum based on commercially available cut points. Patients receive 0 points for less than high-risk features, 1 point for high-risk features, and 2 points for very high-risk features. You then add the values for their NCCN risk group and genomic classifier risk group. If the value is greater than or equal to 3, then the patient is estimated to have a similar prognosis to the STAMPEDE control arm, and so could be considered for abiraterone intensification. If the value is less than or equal to 2, they have a more favorable prognosis, and we would estimate that the absolute benefit of abiraterone therapy would be lower than that observed in the STAMPEDE population.
Based upon the data in your study, should Decipher be used routinely on our patients with high-risk prostate cancer? And what is your message to centers out there who might not have adopted it yet routinely for high-risk prostate cancer?
Currently, Decipher is recommended by the NCCN based on Simon Level 1B evidence. This means it has been validated in an ad hoc way in older prospective trials like the ones in this study. And this can be used to justify the use of Decipher in clinical practice today. However, as you know, the highest level of evidence is Simon Level 1A. And currently, Decipher is being tested as an integral biomarker in NRG-GU009 and NRG-GU010, which have recently finished accrual. Every physician will have their own threshold for what they consider adequate evidence to alter their practice, and those who wish for some additional evidence may wish to wait to see what the results of NRG-GU009 and 010 will show.
As with any major energy oncology effort, we know that this was, of course, a team effort. Before we let you go, I wanted to also give you the opportunity to acknowledge anybody who really helped to make this study possible.
Thanks. Yes, it certainly was a team effort. There are so many people to thank. Firstly, the enormously influential vision of the NRG-GU committee and the chairs during these trials and analyses, including Howard Sandler, Felix Fang, and Paul Nguyen. Also, of course, the folks at Veracyte, including Eli Divizioni, who collaborated with us to profile these specimens. Next, the senior author and the chair of the NRG-GU Translational Sciences Group, Fu Tran. Lastly, I'll close by thanking the NRG Biobank, NRG biostatisticians, and the NRG staff who obviously are critical for conducting such high-quality research projects.
Dr. Patel, congratulations again on this presentation, and we look forward to seeing these results published in the near future.
Thanks so much for having me. I look forward to sharing a more detailed report with the scientific community on behalf of my co-authors.
To give us a broader perspective of this study in the context of the NRG Oncology Translational Science Program, our next guest is Dr. Adam Dicker, who is the Translational Science co-author on this study. Dr. Dicker is also the Enterprise Chair of the Department of Radiation Oncology at Thomas Jefferson University as well as the vice chair of the NRG Oncology Translational Science Committee. Dr. Dicker, welcome to the NRG Oncology podcast. The term translational science has been with us for a long time. While it generally refers to the process of bringing basic science discoveries to patient care in what is coined bench to bedside, there are many ways of doing translational research. One common example is when we discover a gene pathway that might be causing a sickness, and then we develop a medication to treat that disease. When we talk about translational research in NRG oncology, what is the scope of this program, and what exactly do we mean by that?
Yeah, so first, thank you for the opportunity. I was fortunate to get involved in RTOG and then NRG through my first boss, Wally Curran, when he was the chair at Thomas Jefferson. And I benefited from the mentorship of Paul Akunieff in translational science and was fortunate when Paul had too many grants and he had to give up that position in RTOG. I applied for it and was fortunate to be selected. Translational science has changed over the past couple of decades in RTOG and NRG oncology. I'm going to focus some of my remarks to the GU space where Dr. Patel's abstract is relevant. If you go back 15 or 20 years, where the field of science was in translational research was mostly in a single protein biomarker area. And Alan Pollack, who was leading the GU Translational Research Group at RTOG at that time, really was the brains behind taking a very systematic approach with the tumor repository. It wasn't called a biorepository at that time. It was a tumor repository because at that time there were either blocks or slides. So Alan decided we're going to study what are the relevant proteins that we know of at the time. But Alan, to his credit, with a number of other people, was able to really develop a large body of data chipping away at all the various clinical trials that RTOG had at that time. Around 2013, Felix Fang invited Eli Divisione to the GU Translational meeting, and Eli had started a company called Decipher. Eli had licensed what's called a classifier. At that time, there was emerging what are called high-density Affymetrix arrays. What Affymetrix figured out how to do is how to study all the messenger RNAs, transfer RNAs, long non-coding RNAs, and to make it like solid state, to put it on like a chip so that you could isolate a patient's RNA from a tumor block or a slide or a couple of slides, and you run that RNA on this microarray, and you get this readout of the transcriptome of that patient. Eli Divisione wanted to take this transcriptome classifier. It was made up of like 22 genes, transcripts from the Mayo Clinic, refined it considerably, and wanted to have a transcriptome-based approach for prostate patients. And the whole thesis about the Decipher platform is two patients can have the same Gleason score, but their biology is going to be different. You could have two patients with a Gleason 9, and yet one has a higher metastatic potential than the other. You could have two patients with a Gleason 6, which normally we would say is a relatively indolent type of cancer, but it turns out there are some patients with Gleason 6s whose biology is quite aggressive. And the Decipher test was one of the first molecular tests to be able to discriminate between these homogeneous group of patients that we classically use Gleason score.
So, to put that in perspective, when I think about transcriptomics or Decipher, I want to ask you, is it an appropriate analogy? When I think about, you mentioned single genes, single pathways, we're trying to paint a picture of the Mona Lisa, and perhaps your single gene might give you some idea what that picture looks like. But when you have this array like Decipher, that is giving you an expression profile of a wide array of genes, giving you a much clearer idea that, in fact, okay, this is the Mona Lisa.
Yeah, if you want to build on your analogy, Each transcript, which reflects a gene, is a color or a shade of a color. So you go from monochrome to polychromatic, and it gives you greater insight. And it's amazing that from a couple microscope slides, you can scrape off the little that there is of a prostate biopsy, isolate that RNA. And one of our early studies, RTOG, was showing that blocks that were 15 or 20 years old could yield RNA that could be analyzed. It wasn't degraded. With a block, if it's exposed to air, the first couple microns might get oxidized. It's kind of like an old salami. If you shave off, kind of the end, you get to the good stuff. But it was just transformational what it did to the whole organization, and the digital pathology is the next transformation.
On this note, we're speaking about a lot of material that's been stored over the years. There's a famous scene in Indiana Jones, Raiders of the Lost Ark, where the United States government has taken possession of the Ark of the Covenant. And Indiana Jones starts asking some of the officials, well, where is this ark? And one of the government officials simply says, we have top men working on it. And Indiana Jones responds back, well, what top men are you talking about? And then at the end of the movie, we just see the ark being pushed into a big warehouse collecting dust. And when it comes to our treasure trove of biospecimens that we store at NRG Oncology, how do we ensure that these biospecimens are really being studied by our top people and aren't just sitting there in a warehouse like the Ark in Indiana Jones?
I do recall the scene. It's quite vivid in my mind. For a small clarification, so we're custodians. It's not our material. It's really the government's material. There are different biorepositories around the country. Each NCTN group has their own biorepository. The biorepository, by the way, has more than just blocks and slides. It has urine, it has plasma, and it also has digital images, digital pathology. And anyone can gain access to this. So if there was a biotech company that wanted to study brain tumor biospecimens or pancreas specimens or whatever it might be, they would apply through the NCI Navigator website, and there's a process by which those applications are vetted. And there's a small fee that's involved in the shipping and whatnot, and there may be material transfer agreements, etc. But anyone has access to this. We try to promote the usage of the materials in the translational science monthly calls that Vered Stearns and I, and formerly Mike Beerer, have. We promote it at the semi-annual meeting. It's not just NRG oncology investigators that use this material. So likewise, investigators who are, quote, affiliated with NRG, if Alliance or SWOG. I have a collaboration with someone at Dana-Farber in the blood plasma proteomics space. And in that DoD application that we submitted last year, we were pulling specimens from the Hoosier Oncology Group and a variety of other cooperative groups or clinical trial consortium. So this is the American taxpayer is paying for it. It's available. We promote it. Heather Lankeys, who's based in Buffalo, she sits on the group banking committee. And I would say in general, across all the NCTN groups, these resources are underutilized. But the value is the clinical trial data that's associated with each specimen. Anyone in the world can access, and we certainly make ourselves available. And we want people to be successful in applying to the NCI and Navigator process. And the NRG statisticians play a critical role in making sure that the applications to NCI Navigator are high quality. It's a pretty rigorous process.
So, coming back to the study presented by Dr. Patel today, he filled us in on potentially paradigm-changing findings with data suggesting we should be administering patient-tailored hormonal deprivation therapy. This is therapy that lowers patients' testosterone based upon the specific deciphered profile of their tumor, rather than just blanketly giving hormone therapy for all patients with high-risk disease. In the breast cancer space, we've been using a similar approach with things like Oncotype to guide administration of chemotherapy, for example, for decades based upon translational work done in trials like NSABP, B14, B20, B28. This was decades ago. And so, why has it taken us much longer to come up with translational patient tailored strategies in something like high risk prostate cancer?
Kudos to Steve Shack, who 20 years ago, when they came up with the Oncotype DX, so that's a polymerase chain reaction, a PCR-based approach, their approach was we're going to pre-identify genes that we think are important in breast cancer through a massive literature review. They came up with a handful of genes. And then the secret sauce is how much weight do you put on one gene versus another gene to come up with a score. The brilliance of Steve Shack was taking advantage of what the NSABP had, which was a lot of tumor blocks and these randomized trials, and having had the clinical endpoints. So why is prostate cancer taking longer? I think in part is prostate cancer learned from breast cancer about how to do some of these trials in general, therapeutic trials. I think in part it depends on endpoints with the FDA. It took a while to go from a overall survival endpoint to a metastasis-free survival endpoint where bone scans could be used. There was a lot of work with Howard Shear at Memorial Sloan Kettering and many others, prostate cancer working group, working with the FDA so that you could develop endpoints that would be surrogates of overall survival and try to have something that would be meaningful and that it wouldn't take 10 or 15 years to get an answer. I think prostate cancer was slower to get to a molecular classifier. If Genomic Health way back when had focused on prostate cancer instead of breast cancer, then we wouldn't be having this conversation. But I don't think they were wrong, by the way, for going for breast cancer. I think that was a very strategic and smart approach. I'm glad and I'm grateful to the late Felix Fang and the whole community that's gotten us to this point. But yeah, could we have done this 5 or 10 years earlier? Maybe, but the goal is to do the best with what we got.
From a practical standpoint, in addition to your many leadership roles, both at Thomas Jefferson, the NCI, NRG Oncology, you are also an active clinician who treats prostate cancer. And I'm wondering, let's say a patient comes in who's read about this study in the news and they ask you about what it means. Can you boil down what this would mean to a prostate cancer patient in the clinic and how important this is for someone who is facing a high risk prostate cancer, potentially needing hormonal deprivation therapy?
I'll tell you what we're good at right now and what remains to be done. So I think what we're good at right now, or what we're better at, is using sophisticated tools like Decipher or other molecular tests to try to be a little more judicious about who should receive therapy that results in a castrate state in a man, and to know who would get benefit from a cure perspective. This study sheds a great deal of light in that. So right now, when we give any form of therapy to lower testosterone, chemical therapy, drugs, we can't predict the severity of the side effects or toxicity to patients. So it has nothing to do with body mass index. You could be 250 pounds, 6'5", all muscle, and LHRH therapy can result in that person being a couch potato and have significant fatigue. And we don't know who's going to develop hot flashes and who's going to get drenched and who's going to have minor hot flashes. There's a small percentage of patients who still have, for reasons we don't understand, sexual drive, even though you shouldn't have it with these drugs. So there's a concern for long-term administration that there are cardiometabolic effects. And there's no precision medicine approach right now when you have someone who might be a borderline diabetic or with a borderline body mass index, and you're concerned that is the hormonal therapy going to tip them over into an unfavorable cardiometabolic state? So every clinician has this like, well, I think, and the answer is it's total guesswork right now about how aggressive one should be with the hormonal therapy. So vis-a-vis the tumor, I think Dr. Patel's study and others have shed a great deal of light of where intensification of hormonal therapy has benefit from a holistic standpoint and how it affects other organ systems in the patient, particularly the heart, cognition, etc. We're not there yet from a precision medicine perspective, but I think we can get there, and that's one of my interests, blood plasma proteomics, amongst other things.
You had already mentioned that the translational biomarker program, the storage of these biospecimens, is largely funded by the American taxpayer. We are at a time in history where there is a lot of uncertainty over federal funding for things like translational programs, and it's important that we justify these programs to the American taxpayer. Can you give us a sense of what the budget is for translational research at NRG Oncology and how is it spent? And what is the best argument you can make that these studies are a good return on investment?
The major expenses, there's one grant, the major grant that's really for the clinical trial apparatus of NRG Oncology. There's a separate grant that relates to the biorepository that's paying for pathologist time, it's paying for tissue acquisition, cataloging, storing in liquid nitrogen or wherever the appropriate medium is, shipping out tubes for blood collection and other stuff like that. That grant currently is based, Jeff Simcoe for NRG Oncology is PI for that biorepository grant for NRG Oncology, and it supports I think 3 or 4 people at UCSF. The human capital that I and others contribute to the Translational Science Committee, pretty much people are donating their time because we believe in the mission. The ROI is you wouldn't have been able to have a deciphered test that ultimately got commercialized and FDA and CMS approved if it didn't have all that NRG data behind it. And by the way, once they developed it for prostate, they developed something similar for bladder cancer, and that generates, revenue, that generates taxes for the American government. The ROI is multiple fold. There are numerous grants that members of the GU NRG oncology community have received, whether it be from foundations like prostate cancer foundations, whether it be from the NIH NCI, whether it be from pharma. So, there's a flywheel effect that happens from this research, but it's all based on having a biospecimen that's associated with high quality clinical data. And that high quality clinical data is mostly generated in the community by community oncologists who are putting these patients on trials with the associated research structure associates.
Dr. Dicker, I wanted to just thank you again for coming on the NRG Oncology podcast. And I want to congratulate, of course, you and all the investigators on this presentation at ASCO today, which is really the culmination of decades of work from the Translational Science Committee.
Well, thank you for the opportunity. It's a team sport.
Today, for the 2026 ASCO annual meeting, we heard from Dr. Krishnan Patel on results of a major translational analysis evaluating the role of the Decipher test to guide the administration of hormonal deprivation therapy for high-risk prostate cancer, and Dr. Adam Dicker, the vice chair of the NRG Oncology Translational Science Committee, to put all of this in perspective. The NRG Oncology Group is supported by funding from the National Cancer Institute, and content of this podcast does not necessarily reflect the official views of the National Institutes of Health. Thanks everyone for tuning in to this special 2026 ASCO episode, and happy clinical trials.